Okay, let's start our today's topic. So, last time we were discussing about the classification of enzymes. Now you know enzymes, they are classified into different types. First class of enzyme that we discussed was oxidoreductases. You now understand that oxidoreductases are those enzymes which catalyze simultaneous oxidation and reduction of their substrates. Then we discussed another class of enzymes that was transferases. Transferases are the enzymes which catalyze the transfer of groups from one substrate to another substrate. Then we discussed hydrolases. Hydrolases are those enzymes which break complex molecules into simple molecules by hydrolysis, by the introduction of water. Then we discussed lyases. Lyases also break complex molecules into simpler molecules, or they break the molecules without introduction of water. Then we discussed isomerases, those enzymes which are involved in the catalysis of breaking of bonds to form isomers. Now today we are going to discuss the last class of enzymes, that is known as ligases. So what are ligases? These are the enzymes which catalyze. They catalyze the formation of bonds between two molecules to join them together. Join them together. So ligases are those enzymes which join two molecules together by formation of bonds between them. The famous example of ligases is DNA ligase. So what is the function of this DNA ligase? It joins the two cut strands of DNA together in the process of DNA replication. You might have studied DNA replication. In DNA replication, double-stranded DNA, it is first unwound and it is changed into single-stranded DNA molecules by the help of an enzyme known as helicase enzyme. This is the helicase enzyme. Helicase unwinds the two strands of DNA, and this fork, it moves forwards. As the replication occurs, these two strands are unwound on the master strand and on this another strand. The newly formed DNA, new DNA is synthesized. You know, DNA replication is a semi-conservative process. When the DNA replicates, the two parental strands, they are conserved, and on each parental strand, a new stretch of DNA is formed, right? So as this replication proceeds forwards, this unwinding of DNA, it generates torque ahead, and this DNA strand, it becomes highly coiled. To release the torque, an enzyme, topoisomerase, works. Topoisomerase is an enzyme which releases the torque by cutting the DNA strands. So DNA strands are cut by the help of topoisomerase, so that this turning force, this torque, it is released. Then these cut DNA strands, they are again joined together. These cut DNA strands are again joined together by the help of an enzyme that is known as DNA ligase. So DNA ligase rejoins these cut DNA strands together, right? So it joins two molecules together by the formation of bonds between them. So this is DNA ligase, right? Similarly, you might have heard about amino acid tRNA ligase. We have discussed the structure of transfer RNA. You know the function of transfer RNA. The function of transfer RNA is to carry amino acids from cytoplasm to ribosomes. So how transfer RNAs carry amino acids to the ribosomes? So transfer RNA first, this amino acid, it first binds to transfer RNA. How this amino acid binds to transfer RNA? It binds to transfer RNA by the help of an enzyme. Amino acids, they first bind to transfer RNA. These amino acids, they join transfer RNA. They bind to transfer RNA by the help of this enzyme, amino acid tRNA ligase. So by the help of this amino acid tRNA ligase, amino acid first joins to transfer RNA. This is the transfer RNA. We have studied the structure of transfer RNA. This is our transfer RNA. It has got that CCA-OH group here. Here amino acid binds to it. So here amino acid binds to this OH group by the help of this enzyme, amino acid tRNA ligase. Now this charge transfer RNA, then it carries this amino acid to ribosome, right? So amino acid tRNA ligase, it joins amino acid with a particular transfer RNA. Similarly, you might have heard about peptide synthesis. Peptide synthesis. This is also a ligase which joins amino acids together for the formation of a polypeptide chain, right? So ligases are those enzymes which join two molecules together by the formation of bonds between them, right? So the famous examples of these ligases are DNA ligase. Now you understand it joins the two cut strands of DNA. When topoisomerase cuts those two DNA strands for releasing torque, then those two DNA strands are rejoined by the help of this DNA ligase enzyme. Amino acid tRNA ligase, it is the enzyme which binds an amino acid to transfer RNA. Then peptide synthesis, it is a ligase which joins amino acids together to form a polypeptide chain, right? So these were the classes of enzymes. So I think there are 6 classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So enzymes have been classified into these 6 classes, and I have cited example of each class. You have to remember the action of the enzyme. You have to remember the examples of those enzymes, right? So that is all about the classification of enzymes. Now, after this, we are going to discuss about properties of enzymes. Characteristics or properties of enzymes. Characteristics or properties of enzymes. So all enzymes, they show the following properties or they show the following characteristic features. So what is the first characteristic feature or first property of enzymes? All enzymes, they show spasticity. All enzymes show spasticity. What is spasticity? That means all enzymes are highly substrate specific. All enzymes are highly substrate specific. All enzymes are highly substrate specific. So every enzyme has got its own specific substrate. That enzyme will only catalyze that specific substrate. For example, if we talk about sucrase, sucrase is an enzyme. This sucrase, it will only work on its substrate, sucrose. Sucrose. And it will convert this sucrose into glucose and fructose. Glucose plus fructose. So this sucrase enzyme, it will not work on any other substrate. It is only meant for its action on sucrose, right? So we say enzymes are substrate specific. So every enzyme has got its own specific substrate on which it acts. So it carries the catalysis of sucrose into glucose and fructose, this sucrase enzyme. It will not act on amylase. It will not act on amylose. It will not act on proteins. It will not act on lipids, right? So the substrate for this sucrase enzyme is only sucrose, right? Similarly, if we take lipases, they will only act on lipids. Lipases will only act on lipids to convert them into fatty acids and glycerol. They convert fatty acids, they convert lipids into fatty acids and glycerol in a stepwise manner. First, a triglyceride will be changed into a diglyceride plus fatty acid. Then diglyceride will be changed into monoglyceride plus fatty acid. Then monoglyceride will be finally converted into glycerol and fatty acids, right? So lipases, they are only meant for converting lipids into fatty acids and glycerol. Lipase will not act on protein. Lipase will not act on carbohydrates, right? So every enzyme is substrate specific. So for a particular substrate, we have got a particular enzyme, right? So this property of enzymes to catalyze only specific substrates. This is known as spasticity of enzyme. So all enzymes are highly substrate specific. They have got a specific substrate on which they will act. They will not act on any other substrate, right? For example, sucrase will only act on sucrose. Lipase will only act on lipids. Proteases, they will only act on proteins. They will only act on proteins to convert them into amino acids, right? So in this way, we find that all enzymes, they are highly substrate specific. And this property of enzymes is known as their specificity, right? Then enzymes show another property. That is, they show an optimum temperature. Every enzyme has got an optimum temperature for best catalytic activity. Second property of enzymes, that is optimum temperature. Every enzyme has got an optimum temperature. What is optimum temperature of an enzyme? The temperature at which enzyme shows maximum catalytic activity. Maximum catalytic activity is known as its optimum temperature. So optimum temperature is the temperature at which enzyme shows maximum catalytic activity. So that temperature is known as optimum temperature. For most of the enzymes, the optimum temperature, it ranges between 5 degrees Celsius to 40 degrees Celsius. For most of the enzymes, the optimum temperature, it ranges in between 5 degrees to 40 degrees Celsius. Usually, optimum temperature of the enzyme corresponds with the optimum body temperature of the organism. For example, what is the optimum body temperature in humans? In case of humans, optimum body temperature is 37 degrees Celsius. In case of humans, the optimum body temperature is 37 degrees Celsius. So our enzymes, they work best at this 37 degrees Celsius because the optimum temperature of enzymes, it mostly corresponds with the optimum body temperature of the organism, right? So different living organisms, they have got different optimum body temperatures. Different living organisms have got different optimum body temperature. For example, humans, they have got an optimum body temperature of 37 degrees Celsius. So our enzymes will show maximum catalytic activity at 37 degrees Celsius. So for most of the enzymes, the optimum temperature, it falls in between. It ranges in between 5 degree to 40 degree Celsius. However, if the temperature goes high, if the temperature exceeds 40 degrees Celsius, when the temperature exceeds 40 degrees Celsius, at this temperature, enzymes lose their catalytic activity. Enzymes lose their catalytic activity. Now, enzymes, they do not show catalysis above 40 degrees Celsius. So why enzymes lose their catalytic activity above 40 degrees Celsius? This is because at high temperatures, the weak hydrogen bonds which hold the structure of protein, they break. As we have discussed, proteins, they first get primary structure. Then they assume secondary structure by coiling, folding, bending, right? Then they assume tertiary structure. Then they assume quaternary structure. And all these structures, they are maintained by hydrogen bonds. They are maintained by hydrogen bonds. They are maintained by disulfide bonds, etc. Right? So on high temperatures, at high temperatures, the weak hydrogen bonds, they break, and that causes denaturation of enzyme. That causes denaturation of enzyme. Denaturation of enzyme. So enzymes lose their three-dimensional structure. They lose their three-dimensional structure. So when enzyme loses its structure, the enzyme doesn't remain active. So enzyme loses the power of catalysis, right? So at high temperatures, the hydrogen bonds of the enzymes, they are broken, and this causes loss of enzyme structure. And this loss of enzyme structure, it is known as denaturation. It occurs at high temperatures. You might have heard when anyone is suffering from high fever, doctor advises immediate application of cold packs, or sometimes doctor says put him in water when the person is suffering from high body temperature. Why immediate application of cold packs or water therapy is advised? Because at high temperatures, the enzymes, they will suffer denaturation. They will lose their structure. All of our metabolic pathways, they are regulated by enzymes. So when enzymes become non-functional, metabolism will stop and the person may die immediately, right? So whenever any person, whether an infant or an adult, is suffering from high fever, the fever should be immediately subsided. Fever should be lowered by giving some antipyretic drugs. If the antipyretic drugs do not work immediately, right, then the person should be given cold packs. If needed, the person should be immersed in water if the fever is not controlled. Because at high temperatures, enzymes lose their structure. That is known as denaturation of enzymes, and all the metabolic pathways will come to a halt, right? Similarly, at low temperatures, below 5 degrees Celsius, enzymes become inactive temporarily. Denaturation is loss of enzyme structure permanent. In denaturation, the enzymes lose their structure permanently. Once denaturation occurs, then enzyme cannot become active again. But at low temperatures, below 5 degrees Celsius, below 5 degrees Celsius, enzymes get temporarily inactivated, or we can say they remain preserved at low temperatures. So if enzymes are subject to low temperatures below 5 degrees Celsius, the enzymes get inactivated. When the temperature returns to normal, they will again become active. But that is not the case with high temperature. If enzymes are subject to high temperatures, the structure of the enzyme will unfold, and that is known as denaturation. And that enzyme cannot then regain the structure. So that enzyme is lost forever, right? But when enzymes are subject to the low temperatures, they become temporarily inactive. They remain preserved. So when the normal temperature is resumed, they will again start catalysis, right? We take advantage of this property of enzymes. You know, our most of the foodstuffs, they are spoiled by the enzymes of bacteria. When we keep food at our home for 1 or 2 days, that food gets spoiled by the action of bacteria. Bacteria release enzymes, and those enzymes, they cause decomposition of that food. So how we preserve that food? We keep that food, which is meant to be stored. We keep that food in refrigerator. In refrigerator, there is low temperature. So low temperature temporarily inactivates the enzymes. If the bacteria are present in the food, those bacteria would have secreted their enzymes, but those enzymes will not work because of low temperature, right? So the food does not get spoiled. So in order to preserve our foodstuffs, we should store them at low temperature, so that even if bacteria are present there, even if they release enzymes, those enzymes will not work because they will be temporarily inactivated because of low temperature. Then after a few days, we take out the food, we cook it. When we cook the food, high temperature, they spoil the bacteria, they kill the bacteria and their enzymes, and the food is uh, food is preserved. So that is the optimum temperature of enzymes. So most of the enzymes, they are active in the temperature range of 5 degree to 40 degrees Celsius. However, if the temperature exceeds 40 degrees Celsius, enzymes may lose their structure. This loss of enzyme structure because of breakdown of hydrogen bonds is known as denaturation of enzyme. And this denaturation of enzyme, it destroys the enzyme once for all, right? If it happens in the body of a living organism due to high body temperature, all the metabolic pathways will come to a halt and the person may die immediately. Similarly, low temperatures, they temporarily inactivate the enzyme. So we take advantage of this property of enzyme. We can store our food for longer periods of time by keeping the food at low temperature, because low temperature will inactivate the bacterial enzymes. So that does not allow the food to get spoiled, right? But there are some organisms which live at very high temperatures. Those organisms which thrive at high temperatures, they have got heat resistant enzymes. They are having heat resistant enzymes. There are certain bacteria which live in extreme conditions where the temperatures are very high. How those bacteria survive at those places where there is high temperature? Hydrothermal vents where the temperature is high, there are certain bacteria found. So how those bacteria survive in hydrothermal vents? So they have got heat resistant enzymes. Only those enzymes can withstand high temperature. Those enzymes which are not heat resistant, they will be destroyed at a temperature above 40 degrees Celsius. So that is the second property of enzymes. We call it as optimum temperature, right? So every enzyme shows its maximum catalytic activity on a certain temperature. That temperature on which enzyme shows its maximum catalytic activity, that temperature is known as the optimum temperature of that enzyme. So all enzymes will not have the same optimum temperature. As I told you, optimum temperature mostly is correlated with it corresponds with the optimum body temperature. Then we will discuss another property of enzyme that is known as optimum pH. Optimum pH. So what is the optimum pH of an enzyme? The pH. The pH at which an enzyme shows maximum catalytic activity. Catalytic activity. The pH at which enzyme shows maximum catalytic activity. That pH is known as optimum pH for that enzyme. For example, we have got Pepsin enzyme. Pepsin is a protein digesting enzyme which is released in gastric juice. You know it is released in the form of pepsinogen. Then it is activated by HCl into pepsin, right? Then this pepsin, it digests proteins. It digests proteins into peptones and proteoses. It digests proteins into peptones and proteoses. So at what pH this pepsin works best? Our pepsin, it works best at a low pH of 1.2 to 1.8. So it works in strong acidic medium. It works in strong acidic medium. When you go for a feast, after taking a lot of meat, then you take cold drink. You take Coke. You say that this Coke will help us in digesting this meat, this food, right? So how Coke helps you to digest the food? It is correct that Coke helps us to digest the food. How does Coke help us to digest the food after a feast, right? So this Coke, it has got dissolved carbon dioxide dissolved carbon dioxide in it. Carbon dioxide and water, they combine, they form carbonic acid. So it creates acidity. Carbon dioxide plus water, they combine, they form carbonic acid, H2CO3, right? This acid, so coke creates acidity in our stomach. So when pH is lowered at this acidic medium, in this acidic medium, pepsin gets activated. Pepsin shows maximum catalytic activity and it converts proteins into peptones and proteoses, right? So our enzymes, which are released in gastric juice for digestion of proteins, they work best in an acidic medium. They work best at a low pH, right? So pepsin has got an optimum pH of 1.2 to 1.8. So at this pH of 1.2 to 1.8, pepsin will show maximum catalytic activity. If the pH is raised, now if the pH goes to 5, 6, right, 6.5, then pepsin will not show the maximum catalytic activity. The catalytic activity of pepsin, it will slowly decrease, right? If the pH is lowered, the catalytic activity of pepsin, it is increased, right? So when you take coke, that creates acidity in the stomach, and in that acidic medium, pepsin shows maximum catalytic activity. But for small intestine, the enzymes have a different pH. Those enzymes which are released in intestine, they work at a different pH. For example, we have got another enzyme, trypsin. Trypsin is also a protein digesting enzyme. It converts again proteins into peptones and proteoses. So trypsin is also a protein digesting enzyme, but it is released in intestine. It is released in small intestine. It is released in small intestine. So this trypsin, it doesn't work best in acidic medium. This trypsin, it works best in alkaline medium at a pH of 8.0 to 8.5. So some digestive enzymes, they show maximum catalytic activity at acidic pH, for example, pepsin. But some digestive enzymes, for example, trypsin, it shows maximum catalytic activity at a pH of 8.0 to 8.5, right? So it works in alkaline medium. So all the enzymes do not have got the same optimum pH. So this optimum pH of enzymes, it varies. Some enzymes work best in acidic medium like pepsin. Some enzymes work best in alkaline medium like trypsin. Most of the intracellular enzymes, most of intracellular enzyme, cellular enzyme, they work best at neutral pH. At neutral pH, that is 7. So enzymes work in different mediums. So some enzymes work best in acidic medium, for example, trypsin, renin, right? Some enzymes work best in alkaline medium, for example, those enzymes which work best in acidic medium are pepsin and renin. Those enzymes which work best in alkaline medium is trypsin, chymotrypsin, right? Carboxypeptidase, procarboxypeptidase. They work best in alkaline medium. But most of the intracellular enzymes, they work best at neutral pH, right? So every enzyme has got its own optimum pH. So now what is the optimum pH of enzyme? Optimum pH of enzyme is that pH on which enzyme shows maximum catalytic activity. And it varies for different enzymes. So some enzymes will be showing maximum catalytic activity at acidic medium, in acidic medium. Some enzymes will show maximum catalytic activity in alkaline medium. And some enzymes will show maximum catalytic activity at neutral pH, that is 7 pH, right? So that is the optimum pH for the enzymes. After this, we are going to discuss another characteristic feature of enzyme. That is their effect of substrate concentration. Or first, we will see how the enzymes join with the substrate, how they form enzyme substrate complexes. Our time is now running out, so this is sufficient for today. Inshallah, we will discuss that in the next lecture. Thank you for being